So, very good morning to everyone. How are you, children? Am I audible, visible, everything all right? Good morning, good morning, good morning. So, you people wrote the exam yesterday? I am fine. You wrote the exam yesterday? Good, good, good. Very good. So, you did the question. Question was okay, na? You managed to handle everything? Very good morning, very good morning, very good morning. Very good, well done. Good, 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 very good. One student asking what was use of a study exam. The simply use of the study exam is what you studied in the last 1 week, how much you understand, and you revise it properly or not. Means simply to checking your seriousness, preparation. Are you reading NCERT? Are you listening the lecture properly? Getting the things or not? Right? Okay, you joined study, no problem, right? Na? So, no problem at all. Okay, so now we are going to continue what we are teaching. No problem, no problem. Not wrote the exam, not a big issue, don't worry, but try. See, study is a continuous process. If you are in the process, you are reading, you are listening the lecture, you are writing the exam, what will happen? It will help you to grow. It will help you to understand the subject. You just heard a phrase, na? Ki Rome is not built in a single day. Likewise is the education. There is no any shortcut is there. If you really work honestly, if you really work hard for these 2 years, definitely you will got your good rank in the exam. See, around 24 lakh students are writing the exam, and few thousands got the seats. So, of course, we have to prove our edge on the others. And how it will done? Only one story is what basically. Read, where you are finding any problem, discuss with the teacher, getting the things or not. And be consistent, be in the system, be a part. If you read every day, definitely why the seat will not come. Best guide for biology, NCERT, and when you came in the college, we will provide you the study material, so no need to purchase any XYZ guide. Got the idea? So for NEET exam, there are not 3 attempts. For NEET exam, medical entrance, there is one exam conducted in a year. And it's not like any attempt or limit, you can write many times, but most of the good student who qualify the exam in first attempt as well. You see the toppers of the NEET exam. First time they give the exam and become the country topper. So just try to be like this. Very good. Got the idea? College will not provide you the NCERT book, dear. You have to purchase it. College will provide you the study material of the college. NCERT you have to purchase, and it is a very less price book. 200, 150 rupees it will come. Got the idea? So you just purchase that NCERT and start your preparation. Biology, you ask first year biology book NCERT to shopkeeper. They will give you. First year NCERT biology book, please provide me, they will give you. Got the idea? Any NCERT, old or new, doesn't matter. You just purchase it. When you will come, better if you are getting old, it is more better. Right? How many days online classes will continue? Probably it will continue in this month, and in next month up to also some days it will continue. Most probably we will try as long as college is not going to open before that to give you some day relaxation as well. Don't worry about it. Okay, can we start? Can we start now? Next, it is not goal exam. Don't confuse yourself. What study you wrote was not the goal exam. Got the idea? Goal exam will be separate. It is the per week what we are teaching. Na, based on that we are conducting the exam. Listen it. So in a study class, last class we discussed regarding the structure of the bacterial cell. Can we proceed now? Okay. So now study. I think if we start gossiping, a lot of time will go in this only. So what are the main purpose is going to defeat it? We know a lot of question is in your mind. When you will come in college, definitely we will entertain your everything. These are the things. You will got a better guidance. Don't worry about it. Sir, I didn't see starting class. Can I understand the class now? Of course you will understand. Some little difficulty you will got, but don't worry. After that you will understanding. Don't worry. Little difficulty you will got because you are in between now. So connectivity lost is there, but don't worry. You try patiently, listen it, you will understand. Even who started today. Got the idea? Some connectivity will lost, but don't worry. When you will come, we will teach it once again. Have a look now. So we discussed about, as I told you, कि a bacterial cell is surrounded by usually 3 different layers of envelope. I hope you are able to connect it because some new students are sitting. So for them, I am just giving a little background of it. So see, this is a prokaryotic cell. Envelopes. How many different envelopes you are finding in a prokaryotic cell? How many different envelopes you are finding in a prokaryotic cell? Pin your answer. Pin your answer. How many different envelopes you are finding in a prokaryotic cell? 3 different envelopes are there. Okay, very good. So these are a prokaryotic cell. This is a structure of a which one? Prokaryotic cell. So in last class, we already discussed about this outermost envelope. So I just want to know from the student who was present in the last class, can you tell me the outermost layer name? Outermost layer name, very good. It is what? Glycocalyx. And I told these glycocalyx is how many possible type? 2 type I told na, depend upon either it is loose or tough. So I told if this glycocalyx is loose, this is called what? And if it is tough, this is called what? Pin your answer. When it is loose, we called it what? Slime layer, I told in the last class. And when it is tough, it is called what basically? When it is tough, it is called capsule. It is called capsule. Got the idea? Very good. What the name of second layer of the prokaryotic cell I told? Very good. Cell wall. Cell wall. And even I told the chemical composition of cell wall of the bacteria. What was the chemical composition? Help me out. What was the chemical composition of cell wall? So I wrote it is made up of which one? Peptidoglycan. So cell wall of a prokaryotic cell is made up of peptidoglycan. Who attend today the only, don't lose your hope. You are less understanding because you are not connected to the previous class. Slowly you will come in the same pace. Don't worry about it. Got the idea? Right. Okay, ji. So, and these Peptidoglycan is made up of which one? Cross-linking of NAG and NAM. I told na, NAG and NAM. Keep in mind. Now the innermost layer. So about these two we already discussed in the last class. So based on a staining of cell wall, I also told there are how many type of bacteria is there? Based on coloring, we told there are two category of bacteria we decided. That is, one is which one? Gram positive and gram negative. Are you able to connect or not? Or forget? Gram positive and gram negative. Very good. Well done. Now today we are going to start with the innermost layer. So two layer we already discussed. So these are a typical prokaryotic cell. These are a bacterial cell. Most of the bacterial cell have three layer boundary. Outermost boundary is called glycocalyx. Second boundary from outside is called cell wall. And the innermost boundary is called cell membrane or plasma membrane. Now today we are going to talk about plasma membrane or cell membrane. It is also called plasma membrane. Okay, ji. So if we talk about plasma membrane or cell membrane, I am writing only one line here. It is like eukaryotic cell. NCERT wrote like this only. So plasma membrane of prokaryotic cell and eukaryotic cell are similar. It is like eukaryotic cell. Now I will say entire classes about not only one class, probably two class, only on which one? Plasma membrane. But when I will teach eukaryotic cell, simply right now I am writing plasma membrane is like eukaryotic cell. I will say later on composition of plasma membrane. I will say later on what are the structural model of the plasma membrane. I will say later on what is the function of plasma membrane. But wait till the starting of eukaryotic cell. Simply here I am writing only what? It is like which cell? Prokaryotic cell. Sorry, eukaryotic cell. Are you getting the idea or not? It is like eukaryotic cell. Clear? Understood? So when I will teach you eukaryotic cell plasma membrane, you will got a better idea. It doesn't mean say there are no, I already told about the shape of prokaryotic cell here. Right now? So I will give you in the brief in the last. So listen it patiently. Otherwise, when you write in between, so our attention get deviated. So in that way, nothing is going to finish. Getting the things or not? So I will say about the shape in the last. If you asked, I will answer you. Don't worry. So what I am saying, it is like eukaryotic cell. Now I was saying one important thing, that is, it doesn't mean plasma membrane of prokaryotic cell and eukaryotic cell is 100% same. I can tell you 5 difference right now, but deliberately I am not telling. Why? Because it is not your need. It is not the demand of your exam. If I say plasma membrane of a prokaryotic cell, if I tell you extra information, this is the difference between plasma membrane of prokaryotic and eukaryotic cell, you will come in trouble in NEET exam. Because at your level, what is written? Ki plasma membrane of prokaryotic cell is like the plasma membrane of eukaryotic cell. So examiner can ask, कि a prokaryotic cell and eukaryotic cells are similar in their plasma membrane. One of the answer will come. So certain time what happened? Extra information become dangerous for the student as well. That's the reason always I am going to say you one thing. Study only what is the need of the exam. What required in the exam. Got the idea or not? Is it? Right? So now I am going to say about extension of plasma membrane. So it is pending for us. We will explain in much detail about the plasma membrane when I will teach you eukaryotic cell. That is clear enough. So right now I am not telling, simply I am writing one word, that the plasma membrane of prokaryotic cell is like eukaryotic cell. Now I am writing about extension of plasma membrane toward inside, not outside, toward inside of cell. Toward inside of cell. These extension present in prokaryote only, not present in eukaryote. So first extension of plasma membrane is called mesosome. And second extension of the plasma membrane is called chromatophore. First extension is called mesosome and second extension is called chromatophore. Look here in the diagram. You see, this is the plasma membrane. I made some extension. So these extension are called mesosome. These mesosome is not present in eukaryotic cell. It is present only in prokaryotic cell. Got the idea? And the other extension here of the plasma membrane is, I am writing here, it is what? Chromatophore. So whoever the new student got some basic idea about extension, you are understanding little bit now. Got the idea? Is it clear? So I am writing the other extension as what? Chromatophore. Now question arises, these extension are why only present in prokaryotic cell? Why they are not present in eukaryotic cell? When I will teach you eukaryotic cell, there I will not say about mesosome, there I will not say about chromatophore. I am saying about mesosome and chromatophore only in which one? Only in which one? Prokaryotic cell. So see, mesosome I am writing in detail. I am telling you about mesosome. Have a look here. So mesosome I am writing here. First thing, it is an extension of plasma membrane present in the form of vesicle, lamella and tubules. Got the idea? Present in the form of vesicle. Vesicle means this type of a structure. It is called vesicle. Lamella, flattened structure, and tubule when it is making a tube like a structure. It is tubule, and flat structure is called what? Basically lamella. So this is present in the form of vesicle. You can say lamella. You can say it tubule, and you can say it as vesicle. Did you got the idea? So this mesosome is an extension of plasma membrane present in the form of vesicle, lamella and tubule. Now question arises, what is the function of the mesosome? Function of the mesosome. So function number one, it increase the surface area for enzymatic action. For enzymatic action. Other function is DNA replication and its distribution in daughter cell. Daughter cell means when one cell divide, two cells are forming. So this is called parent cell and this is called daughter cell. So every parent cell have how many DNA? One DNA. So these DNA making copy and both one getting one DNA. And that is the reason I wrote DNA replication and its distribution in daughter cell. Got the idea? Other function I am writing, cell wall formation. Cell wall formation in bacterial cell or prokaryotic cell. Another function I am writing, secretion. Secretion of material from the cell. Another function I am writing, respiration. Respiration. This is very important function and usually from these function in exam question is going to ask in NEET exam. Got the idea? Examiner can give which of the following is the function performed by mesosome. They will give you first and third are correct, first and second are correct, first, third, four are correct, and all of these are correct. Such kind of questions going to frame from here. Getting the things or not? Right? Enzymatic action for cellular respiration. For respiration. So, cellular respiration, so it work like, it is analogous to mitochondria. It is analogous to mitochondria. In prokaryotic cell, you people ask na, in very beginning, 5, 6, 3, 4 classes before, you people, many of you, if you are attending right now, you people asked a question, ki, sir, if mitochondria is absent in prokaryotic cell, how production of energy occur? And I told you I will give you the answer of this question at the right time. It is not the right time to answer that question. This is the right time to answer it. This is the right time to answer that question. Got the idea? You people ask, na, sir, chloroplast is not there. Who work like chloroplast? How photosynthesis occur in prokaryotic cell? Mitochondria is not there. How ATP production occur in prokaryotic cell? How Krebs cycle occurs? Some of the students who know about how Krebs cycle occurs, sir. How ETS occurs, sir. How link reaction occurs, sir. Such kind of questions, I hope. Are you able to recall? You people asked to me. You pinned it and I told I am observing you what you are writing, but I will answer at the right time. Is it clear? So who is working like all the function of mitochondria in prokaryote performed by this mesosome? These in folded structure. Got the idea? That is the reason I wrote here it is analogous. Analogous, do you know the meaning of analogous? Analogous means functionally similar. Got the idea? Analogous means what? Functionally similar. Got the idea? They have a different origin, but the function they are performing are same. It is called what? Basically analogous. So who is analogous to mitochondria in prokaryotic cell? Mesosome. So mesosome is doing the job performed by mitochondria. Beside of that, mesosome also doing some more job. Mesosome doing some more job. Do you know in eukaryotic cell, this secretion is a job of Golgi complex. Do you know? In eukaryotic cell, secretion is the, if you studied properly, probably any of you, so you will study later on even if you not studied, secretion is the job of Golgi complex. But here secretion is done by whom also? Mesosome itself. One student asking, sir, second point I not understand. DNA replication, see, this is a bacterial cell. And in this bacteria, one DNA is there. Okay. Now when bacteria divide, bacteria is an example of prokaryotic cell. Two bacteria are forming. Are you getting my point or not? So here in one bacteria, only one DNA is there. So both bacteria will get one DNA, na? So what happened? This DNA making one more copy of DNA. It is circular DNA, na? So these are making one more copy of DNA. So this is the process by which one DNA become two DNA. It is called replication. So DNA replication and its distribution to daughter cell, this is also the function of mesosome. Got the idea? Secretions of many substances, secretions of different protein, different enzyme, getting the things or not? I will tell about the secretion later, don't worry. So did you got the function of mesosome? So once again I am saying about the function of mesosome. What is the function of mesosome? Any enfolded structure, there is a thumb rule, increase the surface area. If anywhere enfolding is there, you keep a thumb rule. Ki enfolding means purpose, increasing the surface area. Let me tell you through a diagram. I am making one pipe. And I am making another pipe. So you better know second pipe is enfolded. Now I am taking a glass of water. Simultaneously I am pouring in both. From which pipe water will come out fastly and from which pipe water will come slowly? From first pipe, A pipe water came fastly, from B pipe it comes slowly because it is highly enfolded. So it have a much higher surface area. If I just make a straight like this pipe become triple of this. Are you getting the idea or not? If I make it a straight, pulling it. Got the idea? Is it clear in the mind? So any enfolded structure have one duty to increase the surface area. If it is in your food pipe, it is increasing the rate of absorptions. More absorption occur from here, less absorption occur from here. Are you getting the idea or not? So enfolding purpose, you got the idea? Got the idea? Now one question came, sir, how mesosome help in DNA replication? So simple answer of this question I am giving you. You will feel somehow awkward. It is not your cup of tea. What is the mechanism of DNA replication? Even this thing you will not study in your undergraduate program. When you go in your medical college, there you will also not study. When you go for your post-graduation program in cytology, genetics, there you will got the mechanism of the DNA replication of the mesosome, how it is replicating. And getting the things. Otherwise, DNA replication, a very basic idea I will teach you. It is a DNA. You know about DNA, adenine, thymine, cytosine, guanine, guanine, cytosine, thymine, adenine, adenine, thymine. Anybody aware about it? This is a DNA. You know about this thing, adenine, thymine, or you don't know? You know. Some students saying no, sir. Many students writing don't know, sir. Got the idea? So see, I will teach you at the right time. So if it is there, how replication occur? Look. Old strand becomes separated. I am giving you a basic idea. If anyone not understanding, don't lose your heart, because one of your friend asked this question, I am answering it. And later on, when you will come in the college, we will teach all basic related to it. So this old strand gets separate, and complementary to old strand, new strand are forming. Adenine pair with thymine, cytosine pair with guanine, guanine with cytosine, thymine with adenine, adenine with thymine, thymine with adenine, guanine, cytosine, cytosine, guanine, adenine, thymine, thymine, adenine. It is called replication. Complementary, what is complementary and all, you will understand later when I will teach. So it is replication. Got the idea? Is it clear in the mind? Right? Vesicle, lamella or tubule. This structure is called vesicle. One student asking what is vesicle. If anything like this, it is called vesicle. If any infolding occur like this, it is called tubule. Any flat surface is called lamella. You got the idea? So it is tubule, it is lamella, it is vesicle. In diagram I made na? In diagram I made it. Have a look here. When I draw it, I made it. See the things. Wait a minute. Here I made it. See the things. Getting the idea? Now we are moving ahead. So mesosome is over. Now we are talking about chromatophore. But before going to chromatophore, I want to ask the function of mesosome from you people. Can you please tell me what the function mesosome is doing? What the function mesosome is doing, please tell me. One function, respiration, good. Second function, increase cellular surface area for cellular respiration. Third, DNA replication and distribution to daughter cell. Got the idea? Next is secretion. Got the idea, right? Cell wall formation. Very good. Very good. Well done. Don't forget it. Don't forget it. Revise it time again and again and again. It will remain in your mind, and trust me, there is a maximum chances either one line or a question came from here. In your real NEET exam, it is that much important. If you know this 5 point only, many times I say my student, NEET is a very easy exam. If you are methodical, if you are studying sincerely, you can achieve even 720. It's not that much difficult exam. Do you know? I am saying you a honest thing. Many times people think what? 24 lakh student. Appear. Selection, if you want good college, if your rank is under 5,000, you will got some of the very good college. For AIMS Delhi, if you are from general category, top 50. You are under 50 rank of the country. Got the idea? If we talk about overall government and private seat in country, it is less than 1 lakh. Both government and private college, less than 1 lakh in country is seats. Is it clear in the mind or not? JIPMER even under 100. For JIPMER, even your rank is not like even 100 you will not got 60, 70, 80. Such kind of a score you will got. What basically even sometime 100 you can got the which one JIPMER type of colleges. Got the idea? 100 type of a score you have to go for. Now what I want to say, but you don't disappoint yourself. In these 24 lakh student, real competition occur between 1 lakh students. Real competition. Out of these 24 lakh students, 10 lakh students even don't know in which chapter what is written. They are simply paying the fee to the governments. So it is the start time. Become sincere. You will got your seat. Got the idea? Sincereness always pay dear. But in your state, for getting government college, out of 720 marks, if you are scoring like somehow 580 type of marks, you will got government colleges. you are going to save at least 1 crore rupees of your parent. Because in private college you have to expend 1 crore rupees. So if you want to save your 1 crore rupees of your parent, which marks needed? Only 580. It is not a big deal. It is a very easy deal. And who are not scoring that marks, their parent, even they want to dream to become a doctor, they are going to expend crores of rupees. And even not from a good college. One student writing in, I am from Haryana. So in Haryana, you can got the seat in your state in 600. Rohtak Medical College will come. For Andhra University, your score must be good. It is like 630, 640 type of a score. Got the idea? So don't worry, you keep a target. Don't ask me from which state what. You simply keep in mind, if my score is in 600 category, I am going to become a doctor from a government college. From any of the state probably. In some state it is somehow more, in some state it is safe mark is 620. So if such kind of a score is there, either from you are from Delhi, are you from Haryana, from Punjab, from Chhattisgarh, from UP, from any other part of the country, definitely these are the very safest score to become a doctor from government college. And got the idea? But why to dream? Dream for AIIMS Delhi. You are young, capable boys and girls. So if you are early starting, why to settle on some of the XYZ college? Think for some of the very good, best college of the country. So your relative, your parent, your friend, everyone proud on you. Make people feel proud on you. Now, for that, what is requiring? Working somehow more hours than others. Got the idea? Working somehow more hours, becoming somehow more sincere than others. And dear children, nothing other than education from a middle class people to make you on the top of the world. The things, what is deciding? Suppose that you are studying in AIIMS Delhi, Andhra Medical College, Zipmar Pondicherry, from good college, and you came to your relative marriage function. Do you know? People will talk more about you rather than groom and bride. They will say, are you finding this girls, this boy standing there studying in AIIMS Delhi, studying in Zipmar? So people is talking less about groom, talking less about bride. People are talking more about you. How differences came in the life you got? Now your distant relative will say, very close cousin studying in AIIMS Delhi. Either how far cousin you don't know, but they will say, they will try to attach their name with your name. They feel proud to attach their name with you. One of my cousins studying in AIIMS Delhi. It is not tough, dear. Nothing is tough. Got the idea? Nothing the tough. This year, I think one of your seniors have been sent to AIIMS Delhi. They're studying like you only. From a very normal family ground, son of a farmer, studying in AIIMS Delhi. When you people came any day, we will call that boys. They will also talk to you. Got the idea? Is it clear? So doesn't matter and leave what reservation, what candidate. You simply work. Even one seat will come now, you will got a seat. Don't worry about it. These all are excuses. This number of candidates sitting, that number of seat is there. I am from general category. That is from this category. That is from that category. Blah blah blah. These all are excuses. Got the idea? So when you came in campus, we will talk about all your relevant doubt. But meanwhile, I told what is the most primary work for me and you both, what topic we are going to enhance our confidence and knowledge. So once again, I wish to return on the topic. May I? Can we proceed? So mesosome, you understand, na? Understand? Okay. So now I am going to come on chromatophore. Okay, children? So nothing is difficult. Keep your moral high. In life, every big success needed some sacrifice. If you want to know, everybody praise me. You are great. Everybody want to talk with me. Everybody just see or attach their name with me. Okay, I am known to her, I am known to him. You have to prove yourself. You have to go with some extra mile than others. Are you agree or not? So small work not give a big appreciation. Your mom told go and purchase vegetable from market. You can't expect a big appreciation. Oh my God, how great vegetable you purchase. How they will appraise you. But you crack Ames Delhi. Everybody in your society, near no local newspaper, your village people, your known one, everyone. You know na, the photo who came in the newspaper, my known is there. Got the idea? So once again I am revising the function of mesosome, then I am coming on chromatophore. Give your attention. So mesosome function here I wrote. Once again see. Function of mesosome, it increase surface area for which purpose? Respiration, cellular respiration. Got the idea? Is increased surface area for enzymatic action. Which enzymatic action? Enzymatic action for cellular respiration. Another function, DNA replication and its distribution in daughter cell. Another function, cell wall formation. Another function, secretion. Another function, respiration. Now I am going to come on chromatophore. Chromatophore. Got the idea? So chromatophore is what? Extension of plasma membrane holding photosynthetic pigment. Holding photosynthetic pigment. Got the idea? It is present only in photosynthetic bacteria. Photosynthetic bacteria. All bacteria means all prokaryotic cells do not perform photosynthesis. Photosynthesis only performed by some of the bacteria. So the bacteria who perform photosynthesis, in that bacteria who is present, chromatophores is present because they are containing, holding the pigment, and this pigment absorb the sunlight. So this chromatophore is analogous to, you write it, use your brain, common sense, analogous to which cell organelle? So what is the function of chloroplast in a plant cell? What is the function of chloroplast in plant cell? In bacteria, that function is performed by chromatophore. Did you got the idea? Understand the idea? So this chromatophore trap the light energy. Energy for photosynthesis. Did you got the idea? Function of chromatophore came in mind? Is it all right? So mesosome and chromatophore both are overs. Got the idea? Now we are moving ahead. Now we are once again coming in the diagram of the prokaryotic cell. So have a look here. Give your attention. Here is the diagram. Just a minute. I wrote a lot of in this slide, so this slide become too longer. Now have a look here, give your attention. See the things. I told in the prokaryotic cell there is a nucleoid is present. Main genetic material is there. It is nucleoid. What I told about nucleoid, I will not say anything now. It is what I told, it is single, it is circular, it is single or double stranded? Double stranded. It is double stranded. Got the idea? And if we talk about it is DNA, double stranded DNA. This nucleoid is main genetic material. Main genetic material or genome. Main genome. Got the idea? This is the nucleoid present in a bacterial cell. So in bacterial cell, how many DNA or chromosome is present? One. That DNA is single stranded or double stranded? It is double stranded. Single means one. Circular double stranded DNA. It have information for everything in the, it control like every activity of the prokaryotic cell. You can say it is working like brain. It is working like brain of the prokaryotic cell. It performing every control coordinations. Getting the idea? Is it clear in the mind? Now beside of that, in prokaryotic cell, some extra genetic material is there. Beside of that, in the prokaryotic cell, I told the name also. These are extra genetic material are there. And these extra genetic material in this prokaryotic cell is called plasmid. I told earlier also, so plasmid is what? Extra genetic material. It is extra genetic material. It is not the main genetic material. It is also double stranded. It is also circular, but it is smaller than main genome. Smaller than main genome of bacteria. Main genome is what? Smaller than nucleoid. I am writing directly. It is smaller than nucleoid. Understanding the idea? So this is what basically plasmid. You got the idea? Got the idea? So plasmid is extra genetic material. Now plasmid have any important things? So plasmid may contain antibiotic resistance gene. Antibiotic resistance gene. So the bacteria who contain plasmid, it may possible they have resistivity against some of the antibiotic. Means we can't kill that particular bacteria from a specific antibiotic. So this antibiotic resistance gene is present in which one? Plasmid. Getting the idea or not? Main genome is essential for a bacterial cell. What is the difference between main genome and plasmid? So main genome is bigger, plasmid is smaller. One difference. Main genome control all the activity of the bacteria. It is essential for survival of the bacteria. If a bacteria don't have main genome, bacteria cannot survive because it is controlling all the function. Plasmid is the extra genetic material. It don't have important role in the survival of the bacteria. If plasmid is even not there in a bacterial cell, survival get not impacted. But plasmid contains certain gene and who help bacteria in some of the adverse condition. Suppose that a bacteria whose plasmid have antibiotic resistance gene, so that bacteria we cannot kill with a particular antibiotic. Suppose that a bacterian plasmid have ampicillin resistance gene. You know na, ampicillin is a antibiotics. Got the idea? Is it clear in the mind? Okay. Is it? So we cannot kill it with which one? We cannot kill it with which one? Ampicillin. One student asking some bold one, sir, then chromatophore is only enough in the leaf, sir, then why chloroplast is there? See, chloroplast doing some advanced functions. So if any specialization is there, they do the work with more efficiency. So efficacy or efficiency of the chloroplast is more than which one? Chromatophore. Who can go for better photosynthetic yield? One student asking, sir, because I am straightforward giving answer. One of your friend is asking, sir, you tell me if chromatophore is sufficient for photosynthesis, then why chloroplast came? For performance of the photosynthetic yield increase with which one? Chloroplast. Got the idea? Is it clear in the mind? Right. So you got the idea about the plasmid. Plasmid is extra genetic material, nucleoid is main genetic material. Nucleoid is bigger, plasmid is smaller. Got the idea? Another thing, nucleoid is the main genome which control all activity of the cell. It is called extra genetic material, not necessary for the bacterial cell. If it is there, it do some help. What some help is there? It provides resistivity against antibiotic is an example. Now I am going to tell what another things present in the bacteria. Two more things I am going to say. One I said in the last class about polyribosome. I told that when many ribosome holded with messenger RNA, it is called polysome. So I am going to take about polysome or polyribosome. Let me write about the polysome. Polysome or polyribosome, both are same thing. Polysome or polyribosome. See, this is ribosome. 70s ribosome present in the bacteria. You better know, I already told you. So this is many ribosome, and these ribosome attach with the help of a messenger RNA. What is messenger RNA? I will say it later. So this structure is called polysome. In prokaryotic cell, ribosome, if I say, ribosome in prokaryotic cell, which type? 70s type. 70s type, and these 70s type have two subunit, 30s and 50s. Got the idea? S, 30s and 50s. So what is 30s, what is 50s, we will teach during ribosome. So ribosome is 70s type in bacteria, and many ribosome, when many ribosome join together with the help of mRNA, with help of mRNA, it is called polysome. What is the function of the ribosome, or you can say polysome? Function is protein synthesis. In bacteria, protein synthesis occurs with the help of ribosome. What is protein synthesis? How protein synthesis occurs? What is the mechanism of protein synthesis? You will study in the second year, even not in first year. What is mRNA? I will tell you in a chapter biomolecule, not right now. It is a macromolecule. Meanwhile, you keep in mind, mRNA is a macromolecule. Got the idea? So ribosome, which type of ribosome present in prokaryotic cells? 70s. It is divided into two subunits, 30s and 50s. Don't think like 50 plus 30 equal to 80. It is not mathematics. Got the idea? What is S? I will tell it later. So many ribosomes, suppose that it is 30s. I am drawing here, it is 30s, it is 50s. 30s, 50s. A smaller subunit 30s, larger subunit 50s. So when many ribosomes join together, it is forming polysome and it is associated with protein synthesis. And now I am going to tell you about inclusion bodies. Inclusion bodies. Inclusion body is what? Basically, I draw a diagram last time as well. I hope you just able to connect it. I draw the diagram very first time about inclusion bodies as well. So I am drawing here also inclusion body diagram. Look here. Inclusion body. These all are present in which one? Cytoplasm. I told na, inside the plasma membrane fluid filled matrix is there, that is called cytoplasm. I told in the last class. So here it is inclusion body I am going to discuss about. I told about this, I told about this, I told about this, I told about this. So I am now telling about which one? Inclusion body. Give your attention. Inclusion body. What is this inclusion body? So it is non-membranous storage granules. It is not surrounded by any membrane. It is non-membranous storage granules. Some of the example of inclusion body I am writing. Example is Cyanophycean granule. Cyanophycean granule. Another example I am writing glycogen granule. Another example I am writing phosphate granule. Phosphate granules. Another example I am writing gas vacuole. So these all are the example of inclusion body, and Cyanophycean granule store starch. Glycogen granule store glycogen. Phosphate granule store phosphate. If you talk about gas vacuole store air. And these gas vacuole help to provide buoyancy in aquatic photosynthetic bacteria. Photosynthetic bacteria. So suppose that this is a bacteria and it is present in water. So here gas vacuole is there. These gas vacuole store air, and when storing the air, what happens? Air provide buoyant forces, so it is present under the water. It is present under the water. So these air storage capacity helping the bacteria to float on the surface of water. This possible due to storing of air in the gas vacuole. It helps to float on the surface of water. Otherwise, these bacteria will sink in the water and they will go near the bottom where light is not reaching. How they do the photosynthetic? So there, this gas vacuole is helping them. Are you getting the idea or not? There, this gas vacuole is helping. Got the idea? Very hungry, sir. Okay, don't worry. I'm leaving you in 1 minute. First, let me ask, what I told today, did you understand? I told today about plasma membrane, mesosome, chromatophore, polyribosome, gas vacuole. These things came in your mind, right? Chromatophore is the extension of plasma membrane who hold the pigment and absorb the sunlight for photosynthesis. Got the idea? Everything clear? All right, understand? Once again, explain inclusion body. Inclusion bodies I wrote, these are non-membrane storage granule. So these are the structure who store The material inside the which one? Prokaryotic cell. They are not covered by any membrane. These are called inclusion body. Example of inclusion body I wrote: synophysin granule store starch, glycogen granule store glycogen, phosphate granule store phosphate, gas vacuole store air. What is the purpose of storing air? Helping to float in the water. Buoyancy means what? Float in the water. When you inhale, take deep breathe in and float in the water, you will not sink. As long as air in your body, you will not sink in the water. Got the idea? Okay, ji, thank you. Have a good day to all of you. Bye bye.